Let's cover the general characteristics of Mycobacterium tuberculosis. It is an obligate aerobe. It means it requires oxygen must for its survival. Without oxygen, it cannot survive. It is an acid-fast bacilli. Bacilli means rod-shaped bacteria, and acid-fast means it resists decolorization with strong acids. As we know, in gram staining, we use decolorizers like acids and alcohol for the decolorization of the stain. But here, this bacilli cannot will be decolorized with strong acids because of the thick mycolic acid layer in their cell wall. Mycolic acid is basically a lipid which is made up of long chain of fatty acids from carbon 78 to carbon 90. So due to this reason, we will not use gram stain and we will use Zn stain instead, because the carbalfusin in the Zn stain can penetrate the thick mycolic acid lipid layer deeply and will stain the bacteria red. The virulence factor of Mycobacterium tuberculosis is the cord factor. As we know, virulence factors are those factors through which the bacteria can cause the disease and spread the disease and survive inside the host. And here, the cord factor is a glycolipid. The most important property of the cord factor is that the virulent strains of MTB, which have the cord factor, is easily identified under the microscope by identifying the serpentine cord-like pattern. Serpentine cord-like pattern is the snake-like pattern. As you can see, it is twisted, winded like a snake is twisted and winded. This is known as serpentine cord-like pattern. You can detect it in the virulent strains of the mycobacteria. Also, Mycobacterium tuberculosis is non-spore forming and non-motile.